welcome everybody, and thank you for your patience. It's as I said, it's a special day. We have Predrag here, no coffee machine, but he will be acting as the caffeine to us.
Yeah.
I think I've known Predrag for a very long time. And when did we first meet? 1999?
I think so.
Something
in
way
San
back
Antonio.
in San Antonio,
Yeah.
where in fact, I remember when we went to I had my first shop, we went to the lecture theater and it said outside, no concealed handguns. And I'm thinking we are really in Texas here, right? So that was a special kind of thing. And in many ways, I'm thankful that those people with handguns, hopefully they need a psychiatrist like you to help them not use those handguns, yes?
Yeah, maybe we need to change the system as well.
Maybe indeed, yes. But what you're going to talk to us today is something you've been working on for a very long time.
Right.
You've been thinking about this particular topic. So I feel very lucky that we have the possibility of actually hearing your new ideas. For those of you who've been following this series, you will know that they also are very closely related to what we heard from Shamil. So for those of you who are unfamiliar with the prediction and the prediction error framework, I strongly encourage you to watch Shamil's talk because he explains the Bayesian brain so well. So in many ways, you probably don't need, no. So you need to see this before you can do this. Yes? No, but thank you so much. It's a real pleasure to have you here and hear about you talking about delusions in the brain and what should do about noise. So please, Predrag.
Okay. Thank you for inviting me. It's a great pleasure to be here today. And I'm giving a talk now, as Morten said, on delusions and the brain. And it's a very much updated version. So you are kind of the testing crowd here. So we'll see how much I'll change it for next time. Anyway, so my background, as Morten said, is in cognitive neuroscience, but also very much psychiatry, clinical psychiatry. I work as a psychiatrist with psychosis patients, and I can tell you that they teach me more than all the textbooks in the world. So anyway, so this title is of course taken from Shakespeare, and the idea here, I think it will be self-evident in the talk why we have this title with a slight change where we change nothing to noise. Okay, so we need to set the stage. So first of all, I would like just to give a brief introduction about psychotic symptoms that you see in psychosis patients, for instance, schizophrenia patients, but also in the general population, which is quite interesting. And I have these, let's say, the more the larger explanation of symptoms later on if someone's interested, because these symptoms are very fascinating, and the different variants that you can see in the clinics. However, if we just have a more core picture of the symptoms in psychosis, they are divided between hallucinations and delusions, but also sensory and perceptual aberrations. Apart from that, we have cognitive dysfunction, negative symptoms, disorganization, catatonia, that this particular lecture won't touch much upon. So just a quick mention of hallucinations. So I won't talk much about hallucinations. I just want to say that this particular field of research on psychosis has been quite well studied already. And just if someone is interested, there are two quite fresh articles in Science that are just a couple of years old that try to understand these symptoms from a predictive coding perspective that we also have that I will also have here in this lecture. And one of them is written by Katarina Schmack, that was a collaborator in some of the studies that we've done, and she actually does the same experiment both in mice with optogenetics and in humans. That's quite cool. Anyway, also there has been quite a lot of research on sensory perceptual aberrations. So if you're interested in this, you can read a review article on the sensory processing dysfunction in psychosis. I will actually touch more upon this as a part of the talk. And when you think about delusions, some aspects have been studied. Sometimes, at least in the clinics, we say that two things belong to delusions, which I don't really agree. So first of all, it's about ownership. Who owns your body? That you have this experience that you are actually the owner of this body. And maybe even more importantly, agency. Who is actually controlling your movements? Is it you or someone else? Well, often in schizophrenia, these patients can have experience that they lack this control and someone else is doing this. And actually these agency and ownership can explain a lot of more also cognitive thought symptoms that you can see in these groups. And this has been quite well studied also in terms of the underlying mechanisms. However, when we think about proper delusions, what people in common think about when they think about delusions, like paranoia, but also like ideas that are not, that no one else would accept as something that is in reality. This has not been studied that well, and the underlying mechanisms have not been studied that well. And what is so fascinating is that this is not only seen in our patients with psychosis, it's also seen in the general population. I don't know, this article in the Atlantic is some years old right now, but it's still so beautiful, so I can recommend you to read it. So so this was before Donald Trump and before all this happening, but it gives you a historical expose about psychotic symptoms in America. And the numbers of these types of symptoms in the population, in percent, they are, I seldom use this word, but here I need to use it. They are flabbergasting. Look at this. So this is just from this article. So basically, 33% believe that pharma industry and governmental bodies are hiding effective treatment against cancer. Almost 25% believe that vaccine causes autism, and this was before COVID pandemic. 50% believe that media and government add secret mind-controlling signals in TV broadcasting. That's a hardcore one. 25% believe that witches exist. 25% believe that American agents were behind the 9/11 terror attack. So this is really, and I haven't checked these figures, but this is just what they wrote in the Atlantic. But I know that similar figures are shown in research for other types of beliefs. Okay, so we can actually measure how much delusion-like beliefs there is a population. There are standardized ways to do it. So, for instance, we use a questionnaire that is called Delusion Proneness Questionnaire, which is actually asking questions that are similar to like what patients have in delusions, but a little bit nice, a little bit more, less intense kind of. And as you can see, there is almost a normal distribution when people try to answer these questions. And what you have to understand, these are not patients. This is us. We are all somewhere on this line, okay? And the thing is also, if you have high delusion proneness, it's not just bad. I mean, there is research suggesting that, for instance, creativity is kind of associated with this. But it can also get you in trouble, and it can get the whole society in trouble. And also, there's another problem. If you have high delusion proneness or psychosis proneness, more general, there is a larger risk that you can develop or will develop a manifest psychotic disorders such as schizophrenia. So I think if you have high delusion proneness, maybe the risk is 10% instead of 1%. Most people don't, but some will. And especially if you're exposed to stress and drugs and similar. All right, setting the stage. Number 2, pathological processes underlying schizophrenia. What you have to understand, schizophrenia is not, as they said before, a disorder of the soul. schizophrenia is hardcore biology happening in the brain. There are mechanisms that underlie that the brain is actually processing information in a different way. And we have now studied, for instance, the genetics of schizophrenia for many years. And the problems that they showed in these studies are that they were not able to identify like 10 genes that are actually that are contributing to schizophrenia. Instead, it seems that it's like the total score of many different genes that together make up the risk. And you can actually today define polygenic risk scores. So there are like 130, 150 genes that we believe together kind of can increase the risk. And you can actually also make studies from, and I will show you later at least one study which has actually used the polygenic risk scores to kind of to understand what is happening in the brain. All right, so there are several different hypotheses that are for what is happening in the on a fundamental level in the biology in relation to schizophrenia. So synaptic pruning hypothesis, I think, is very, very interesting because it actually looks at the fact that there's a lot of things happening developmentally from the age of 5 to the age of 25 in the brain. And for instance, the prefrontal synapses will excitatory synapses will go down quite a lot, while the prefrontal inhibitory synapses will go up quite a lot. But if you look in schizophrenic populations, if you have the same curves here, you see that their synapses, they have much, they will end up with much lower density of all kinds of synapses in the brain during this developmental stage. And this is really interesting, I think, from an information processing perspective, because it actually means that the brain must process the information differently as well, just because it has a lesser connectivity. The synaptic pruning hypothesis is very interesting in relation to some research that is being made right now, and that is actually targeting the human immune system. And we have a fantastic research in Sweden on the immune system and psychosis. I will just give you one example that is looking on the immunohypothesis. This is a colleague of mine whose name is Carl Sellgren. And what he actually does is that he collects dermatocells from patients with schizophrenia, and especially those that have this complement factor C4A, a genetic risk for that factor. And then they do different types of they grow these cells, and they make them turn into microglia. And then a microglia in the brain, it actually will eat synapses. This is what is pruning. It's that they are actually they're actually reducing the number of synapses in the brain. But the microglia of these patients, they're doing it much more. So that can be one of several different explanations why you end up with a system with a lower concentration of synapses. Another hypothesis, the glutamate hypothesis, which actually says that there's a relation between excitatory neurons and inhibitory neurons, in that the inhibitory GABA neurons, they will actually sense how excited the excitatory neuron is, and thereby they will suppress it a little bit, so there's a balance. However, it seems as the NMDR receptor doesn't work properly, and because of this, the GABA receptor cell, won't get the signal from the excitatory neuron, and therefore the excitatory neuron will actually be much more excitatory than it should. So that's another hypothesis. And lastly, I'll show you the hypothesis that I think is most common, the dopamine hypothesis. And this is, of course, that the dopamine system doesn't work properly in schizophrenia patients. And basically what we can see is not that the number of dopamine receptors are different. However, they're more, they respond much more than normal dopamine receptors, and that can be really nicely, that can be shown both way if you measure dopamine synthesis and dopamine release using a small dose of amphetamine in patients versus controls in a PET scanner of the brain. And of course, the dopamine system is one of these neuromodulatory systems that is so important, especially for salience and rewards. And this has developed many different hypotheses how that this can actually induce psychotic symptoms. All right, I'm still setting the stage. So how can we study formation of beliefs from a psychological or cognitive perspective? So if we want to do that, we somehow have to define belief. And in this case, I am not defining belief as a mathematical predictive coding thing, which is usually used. I more try to define belief as a kind of common language idea of belief. And if you look, for instance, in Britannica dictionary, you see that it will say, so it's a feeling of being sure that someone or something exists or that something is true. It's a feeling that something is good, right, or valuable. It's something that a person accepts as true or right, a strongly held opinion about something. But also I would like to add to this that there's also a conscious part about belief. I mean, we at least in our common sense language, a belief is something that we can talk about and discuss and try to understand from different points of view. So because of this, it is conscious. It is also often very complex and is also very abstract. Okay, so how do we study beliefs? I have actually, this has been one of my research lines that I've been interested in since many years in terms of studying the placebo effect. And the placebo effect is really interesting because it's about giving, the sugar pill or a non-active treatment, and it will still, you will still see a result of that. So, for instance, it actually, I mean, it was popularized with Henry Beecher, who during the Second World War, when they didn't have enough of opioids to give the soldiers who were wounded, but then they actually discovered that if they just gave them saline, a third of them would be totally or very much alleviated from their pain. So it actually, there was something here about just the mere kind of giving the treatment that actually induced what we call pain analgesia, so reduced pain. But the placebo effect has been implemented for many years before. So, for instance, you can see the first placebo-controlled trial was in the kind of the conflicts with the Huguenots in France, and there were, because in these old conflicts there were some people who claimed that they were obsessed by the devil, and other priests and some other important people said that they were lying. So they actually tested this by giving them, by telling them sometimes that this is sacred water, and then they threw it on them. And then sometimes they knew this was sacred water, sometimes it was not, and they could see if they were lying or not. So the idea of how you can construct a placebo-controlled trial is actually nothing new. So what about the mechanisms or the processes that are underlying placebo? Well, actually, it's quite interesting to be part of this because we started very early, but it's become like a huge research field of its own. There are even meetings only on placebo research now happening, international meetings that are very big. So people are super interested in this. When it comes to placebo analgesia, that is kind of the reduced experience of pain when you get a placebo treatment, you can see that some regions more often come online. So like the rostral anterior cingulate cortex in the mid-frontal part of the brain, the lateral orbitofrontal cortex, so it's the lower prefrontal part of the brain, and the brainstem. And there are two stories from these studies, I think. So first, there's this the opioid story. And the opioid story in placebo analgesia says that the underlying opioid endogenous opioid system seems to be involved. And we could, for instance, see that there's an activational pattern during placebo treatment of pain that will overlap quite a lot with when you are given opioids, especially in one of these regions, the rostral-anterior cingulate cortex, so the mid-frontal part of the brain. And also it will correlate in a similar way to regions in the brainstem that also have very pronounced opioid networks. In the second picture or image here, you can see opioid receptors in the brain. As you can see, they are very much abundant cortically in the rostral-anterior cingulate cortex, maybe even more so subcortically. But cortically, you find them here, and you find them in anterior insula, basically. And they have even done functional receptor imaging with PET, and they can show that there is actually there is actually opioid receptor activation or indications of that when you treat someone with placebo. So that's the opioid story. So we know the opioid system is activated. But what I think is really fascinating is that there is actually a non-opioid story. And this is, we did a study, this is so old, this is 50, no, what is it? It's 18, 20 years old, very old. Anyway, it's so old, so it's good, because we actually didn't use fMRI, we used PET. And PET is good because instead of having studying events, you can study processes that happen for a whole minute. So so you could see regions that come online and are active prolonged time. Sorry. And the interesting thing is that although we observed these relations in the anterior cingulate that I showed before, that overlapped between opioid treatment and placebo treatment, we observed activations in the lateral orbital frontal cortex that were specific to the placebo treatment. So which is really interesting because it debunks some ideas about placebo. So for instance, that it's a linear effect. It can't be linear when it's not the same thing happening in the brain. There is something happening in the placebo treatment that is not happening in the active treatment. And this is really fascinating if you think about it, because you should have placebo in the active treatment as well. So there's something non-linear that seems to happen there. And as you can see, we also observed a correlation. This region had a increased functional connectivity with rostral anterior cingulate cortex, the other region just talked about, only in the placebo condition, but not in the opioid or the control conditions. We performed another study that was a little bit similar also after this, where we studied emotional placebo. And basically we also here observed, this was fMRI by the way, also here we observed a similar kind of activation pattern in both the lateral orbital frontal cortex and in the rostral ACC. And we also observed a connectivity between these regions that I spoke about. So it generalized these effects a little bit. And since then, people have tried to generalize similar effects as have been done in placebo studies. So, for instance, instead of suggesting that something something should have less pain, you can suggest that something should have more reward, and that has been done in several studies. So, this is a quite famous study that looked at how people react when they sample wine in an fMRI scanner. And they think that some wines are extremely expensive and others are not. And every time it's like the medium type wine that is somewhere not in the middle that they're given. And what you can see, once again, the lateral orbital frontal cortex is activated more, and note it's a little bit more in the medial part of this area this time. And it's also related to the extent of this experience. Another study looked at smells and compared, and this was an artificial smell, but it was kind of stinky. But in some cases, the subjects were told that this is a very nice cheese. I think actually, I think actually it wasn't this one. It was Maybe everyone doesn't think this is a nice cheese, but either it was a nice cheese or smelly socks. But it was exactly the same smell. And once again, you could observe similar activations in the orbitofrontal cortex. Okay, so if this is about suggestions, then it should be even possible to do different types of experiments where you use higher order suggestions. So for instance, one thing that has a similarity to placebo studies is instructed learning. So instructed learning is it's a fear learning paradigm, which is often where you get information before, maybe, so often you have a fear conditioning procedure, but before this procedure you will get information about which stimulus will actually give you a shock and which will not. And then you can compare it to fear conditioning that doesn't have this information. And in these types of studies, you also often have people also often see an increased activation in similar areas. And finally, another approach to study this is cognitive reappraisal. And in these types of tasks, you show different types of pictures that can be emotional or neutral. Often negative emotions are shown. And your task is basically that you yourself should have changed your concept, your idea about this picture. So usually when I show this crying woman, the idea is maybe to tell yourself that yes, this woman cries, but why is she crying? She's crying because she's so happy because her son is back after being away for a whole year. And suddenly you experience the picture a little bit different. When you do these types of experiments and you maybe compare it to distraction, as in this study, you also often see increased activations in these areas. And all of these, so placebo experiments, reward suggestions, instructed learning, and cognitive reappraisal, they are all about higher order information that somehow instruct us, or where we instruct ourselves how we should experience a stimulus. Okay. This has been kind of synthesized and reviewed in studies like this one, where it's actually suggested that these regions, these networks are the neural basis of emotional regulation, which of course may be true, but maybe we shouldn't stop there. Maybe we should try to understand it even in more detail. Morten, of course, did this study or has done a lot of different analyses or meta-analysis and similar on activations in the brain. This is also in the orbitofrontal cortex, and basically he shows that the relations to reward and positive outcomes are more in the medial part, while relations to punish or negative outcomes are more in the lateral parts. So this is a little bit in line with what I show you with the mind studies versus the placebo analgesia or emotional placebo studies. Okay, but this is not the full story again, because we tried to make a placebo-like experiment with a completely different, with using completely different stimulus than emotional stimulus. So we used random dot kinetogram which is illusion. This illusion is basically that you see a lot of dots on a computer screen. Those dots will, you will have the experience that they rotate to the left or to the right, but this is ambivalent, so it actually your experiences jumps between these two rotation experiences. But what you can do is actually you can do this non-ambivalent by, for instance, in these using these dots, make them red and green, and using 3D glasses. And then you can really, the experience will be exactly like, so you can predetermine the experience that most people have. So people got these glasses and they thought, okay, great. So they were told that, okay, if you have the red glass on your right eye, the rotation will be experienced more to the right. If you have the opposite side of the glasses, you will have the other rotation. And really that is what I saw. Now, after that, they had a 5-minute break. And just to tell you, this is exactly how we do placebo analgesia studies. We actually give them a lower pain when we give treatment, often first to condition them for the effect. Now, after that, we did the same. They looked at the same random dot kinetogram as in the start, when you didn't have any 3D cues, but they had the glasses and they had the same experience, and they still experienced that the rotation was more to the right side if they had the red glasses on the right side, and vice versa if they had the green glasses on the right side. Okay, so this is an effect that can only be explained by the suggestions that we've given before. So in a way, this is a way to move away from the previous placebo studies that have been done and show this is much more general phenomenon. It's about expectations. It's about the models you have in the brain. And why do we have these models in the brain? Why can't we just experience like the world is? Just, I want the flow of the world. Well, the problem is that often we have so little information. So we need to help our systems to understand the world. So this is a classical, this is a Swedish psychologist that done this like 50 years ago. It's a classical video that shows that only using 24 reflexes you can build a whole story. I'll show this to you in a sec. All right, so you can see that it's a man and a woman. They're in love. They hold hands. They're walking down a beach. I can see a palm and a sun and the sun going down. Can you see it? I mean, we build all these models, although we have so little information. And so this is one reason why we have it. But also, these models that we build are actually help us to learn things. And this is basically what we do in placebo. So as an example, I'll show you this. Has anyone seen this? This was out in social media like 10 years ago or something. I picked it up immediately. So if you see this and you have the question, so what do you think it is?
Oh.
Anyone here? Families?
TV shows. TV characters, yeah.
Okay, I would say there is a DNA. I would say that this is kind of a dad trying to explain the DNA system to a son. But you were actually a little bit dry because look at this. Yeah. I actually looked for these myself, but they were so exactly perfect in the matching for the figure I had. So look at all of them, and you see have you looked at all, compared all? Okay, because now when you see this, you will have different experience. Okay, so I have actually manipulated your brain a little bit. Okay, so what is actually happening here? So even before predictive coding and these ideas were discussed in placebo literature, we actually said this, which was a very similar idea, that you have treatment, you have a treatment expectation in the brain of what will happen, but you will also, in placebo analgesia, for instance, this treatment expectation is that the pain should not be as painful as it was before, but you also will have nociceptive or pain signals reaching the brain, and these will be compared. And when they are compared, the brain will produce an error signal. An error signal will tell the brain something is wrong. I have not understood the world perfectly. And in order to optimize this, you want to minimize this error. And the way to do it is either to change your treatment expectations, or actually to change how you process the input signal. So for instance, use top-down regulation and your opioid system and actually modulate how the nociceptive input is processed. And so one idea is that treatment expectations, at least on the higher order level, like that are mediated by suggestions, etc., that orbitofrontal cortex may be important for this. And anterior cingulate cortex with all these opioid receptors maybe is important in this top-down regulation. At least an idea. After this, of course, people start talking about predictive coding in relation to placebo. And this is just a very simple, this is from Christian Büchel's review paper on this, and it's just a very simple idea of what is actually happening in predictive coding terms, and it's in using Bayesian mathematics. So basically you have, so on the x-axis here you have the ratings of the pain, visual analog scale ratings. So, and these peaks, these different peaks you have here, this is different type of information. So basically the observation, that is the nociceptive input. The prior is that will kind of represent your expectations. And the more narrow these are, the larger is the precision, so the more important it is for how we actually will experience this. And the brain will, or using these Bayesian statistics, then you can produce a posterior, which is kind of a compromise between these two. And the idea, and that's shown in the green. And the idea is that the posterior is mostly associated to our real experience of the pain. And as you can see then in up here, when you have a control condition, the prior is very flat. You have no kind of concepts, no of how it will, what you will experience. And the observation and the posterior are basically overlapping. But if your prior is a little bit more precise, you will see that the posterior will move towards the prior, and our experience will be less pain. So that is actually the idea of how you will get the placebo analgesia. Of course, these ideas are taken directly from all this work that I think Carl Friston has represented well, and this is some of his kind of summary. That, of course, we'll go through all the maths now for 3 hours. No, but the important thing here is actually what Carl is saying here. A part of this is very simple. This is what is actually happening. There are comparisons, and there are comparisons on different hierarchical levels. And this can be, I think, summarized even more simple so you understand it. So basically you have the signal to the brain. The signal will be compared to a low-level prior. If they don't match, so this is what we talked about before. If they don't match, there will be an error signal that will move to the next level, be compared to a little bit higher priors, and so forth. So this is the idea how we could actually use this predictive coding system to process information in the brain. By the way, do you remember this? Do you still see what I... Okay. But this is still not the full story. Okay. Because there is another super exciting literature about the lateral orbitofrontal, or the orbitofrontal cortex, that I want to share with you. So there's a set of experiments that Moshe Bar has done, for example, on object recognition that I love. They're really beautiful. And what he has, in several of his studies, what he has done, he's a kind of a visual expert, perception expert. And what he's done is basically that he has shown pictures very, very, very fast. They're also masked, both before and after the picture. There's coming something that is blurry, that you can't, that makes it even harder to see this picture. And they're repeating, so some pictures are repeated like 5 times, and you maybe don't see what it is the first time, but maybe we'll see the 3rd or 4th or 5th time. But what he can show is something super interesting. First of all, you have activation of the orbitofrontal cortex and of the fusiform gyrus, here and here, using fMRI. But what he also shows is that you have, using MEG, is that the signal in the orbitofrontal cortex precedes the signal in the fusiform cortex. And the signal in the fusiform cortex is actually most activated when you consciously perceive the picture. This is really cool. And he can also show that he can also suggest that these regions, orbitofrontal cortex and these visual regions, are functionally connected as well. So the idea he has is that actually, so you have like you have the early visual processing of information, which will slowly move in the ventral visual stream to the fusiform cortex. This processing is quite slow, but it's very detailed. However, then you have a low spatial frequency part of the picture that we quickly go to the orbitofrontal cortex. And then the orbitofrontal cortex will somehow aid the fusiform cortex to interpret what we are actually seeing. In order to not only say this as a hypothetical thing, he actually tested it as well. So he showed pictures that either had a low spatial frequency or high spatial frequency, and basically it will show that the low spatial frequency pictures were the ones that were activating the orbitofrontal cortex. And not only that, there has been some other people doing similar studies afterwards. So this study, for instance, I'm kind of intrigued by that, because they studied actually what happens when you have a notch. You have an idea, you see something, but you can't tell what it is. And actually at this stage already, you have the activations in the lateral orbitofrontal cortex. A good colleague of mine called Morten Krielbach and his team have made some beautiful studies with babies. And I think it's fascinating because in those studies it seems that you see a similar activation. You also see, using MEG, you see activation in the orbitofrontal cortex that precedes the fusiform activation. And we will be back to that soon. But just want to tell you that later on, Moshe Bar and colleagues, they went on with these ideas and actually showed both in theirs and other studies that it's maybe not kind of that the orbitofrontal cortex chooses among a set of specific items to help out to interpret what we see. Maybe it's actually more about context. Maybe it chooses the context. So the classical thing, Morten and I have discussed this. So is this a pistol or is it a hairdryer? Actually, it's a figure, but is it a pistol or hairdryer? You know, so it depends if it's a bathroom or a war scene, right? So if it's a bathroom, the context is very different, and you will more likely interpret this is a hairdryer. Okay, leaving Moshe Bar and his fantastic work, there's another set of great work that has been produced recently, especially by, I think, Yael Nev has done some really beautiful work that she's also a little bit touching upon similar ideas, but from a completely different perspective, much more computational, and also looking at decision making. Basically, she asked herself, so, okay, we can perform all these different computational processes, like different type of learning, etc., that we can define. However, in those experiments, the setting is always defined. But what is actually defining the setting, or what should be computed in the brain? And this is a really hard task, because we need to know what to compute and what not to compute in order to do it. And basically the idea is that the orbitofrontal cortex is actually have a hidden state representation that is used in decision making. So in this task, for instance, this is one of these classical experiments where you have superimposed houses and faces. I don't know how many you have now, maybe 100 or something, but it's been used for many different things. But in this case, you were supposed to categorize, so you saw young faces, old faces, young houses and old houses, so modern houses and old houses. And you were supposed, and you saw a lot of these stimuli, and the task was to say if there was a young face and another young face, you should go on. But if there's a young face and an old face, you should switch to houses instead, and judge if the house is the same category or not. Okay, so you actually need to think about what has happened before in order to solve this. And basically what she shows, when you go through all these states, She showed, using multivoxel pattern analysis, she showed that the orbitofrontal cortex is very much representing these different states. So here you see it a little bit more on voxel basis. So the previous category is represented, the previous age is represented when you need that in order to solve the task. But this is why she says it's the hidden state representation. But actually also current category is represented. And she says current age is not represented, but actually there is a tendency here as well, and it's actually overlapping almost with previous age. So I think it's both previous and current states that are represented in decision making. But it's the states. And that's the important part there. So you have so this kind of, if you synthesize all these ideas together, we land on a state representation of what I would say global low spatial activation patterns. Can't even say it. So just to recap, so you have the placebo effects, but here I also include other suggestion effects, instructed learning, cognitive reappraisal. You have object recognition, and that is all about expectations. You have object recognition, which is about context. You have decision making, which is about hidden states. And so in placebo research, we say the expectations represent the orbitofrontal cortex that impact low-level information processing through a predictive coding mechanism. The literature, that's the idea in the research. The studies on object recognition and ideas there would say that context with a low spatial resolution is represented in orbitofrontal cortex and aids object recognition. And finally, the work by Linacre and colleagues says that there's a state-dependent decision-making where hidden states are represented in orbitofrontal cortex that aid learning and decisions. All right, I showed you this picture before. I just want to just say a couple of more general things about that. So this is very interesting because this is really how the brain is constructed and how we can think about it. So already in the eye, you have both cone cells and rod cells. So cone cells, those are those that are in the fovea, and the fovea is so small, so you can barely see it. It's here. This is what, and this is where we see details with. This is our only input for details, actually, and the cone cells are probably, it is believed that they are the input to to the parvocellular ganglion cells, and which have small receptive fields. And the cone cells are also interested in details and colors. The rod cells, and there are much more than this, maybe up to 100 million rod cells, and they are all over the eye, and they are believed to be the main input to the magnocellular ganglion cells, and they're interested in global features, black and white, they can't see colors, and they are light sensitive. So already you have this huge division, basically with, I would say, I don't know, this is just a guess, maybe 98% of the retina, you don't see details. You just see global features, basically. And this is shown in what people are usually discussing, the dorsal visual pathway, which is also called the magnocellular pathway versus the parvocellular pathway. And usually when this is discussed, there's the ventral visual pathway is where we used, it's thought that this is what we use to see kind of detailed information, and so including faces and houses, etc. While the magnocellular pathway in the parietal cortex is more about position and how things are spatially in relation to each other. But however, there is a really interesting other transmission with the magnocellular pathway that goes directly to the orbitofrontal cortex as well. So this is kind of what we are possibly using in these types of tasks. Another thing about the orbitofrontal cortex, it's really important to acknowledge that all perceptual sensory dimensions are represented here. So we've talked about vision quite a lot, but all are represented. And also the idea is that it seems that the representations are more global. And this is, in fact, now I'll return to these fantastic studies on babies. This is shown similar studies as I showed you before, when you look at faces of babies, can be shown for when babies actually cry. And this will also have a similar activation, both of the orbitofrontal cortex and the auditory cortex in this term. And if I understand, you can ask Morten afterwards, but it seems here they were not able to show that the orbitofrontal was before, but that is still to come, more experiments about that to understand that better. But at least a similar idea that you have the orbitofrontal cortex is activated extremely early in these processes, in a different type of perception. All right, so I think we land in a very fascinating landscape of global low spatial representation in the orbitofrontal cortex, actually. This is like, for me, this is like, wow, it's a completely different thing happening here in the traditional cortical representations of perception. So basically, if we just summarize what we said before, so the state representation of global low spatial activation patterns would say that there's a global but low spatial representation of the world that includes all information dimensions in the orbitofrontal cortex. There are two information processes stream. One is concerned with global in the brain. One is concerned with global features, and that is the orbitofrontal cortex. And one is concerned with detailed features, that is classical exoreceptive and interoceptive processing streams. And that the global system seems to impact low-level systems processes through priors in order to minimize prediction errors. And of course, a lot of you have seen these ideas that were developed by Bars and Dehaene on the global workspace broadcasting in the brain. And basically, it's when you have input from different streams that suddenly co-activate that you get a conscious experience. So a lot of work has been done by that, also by Morten and Gustavo Deco. There's let's say, an ignition of these networks when you get this the experience, and there are also a specific network that is involved in this, and that's the global workspace. In gray here. And if we would add this to our reasoning, you could say that maybe if you add orbitofrontal cortex to this, maybe you would have like two systems. So one is the general global the networks activated by the global workspace. Another one is like is hidden and present state representations of global spatial global low spatial activation patterns in orbitofrontal cortex. And that this is actually, I blurred it here a little bit, because it should be blurred. And also, this would be, if we go back to the ideas I showed you about the hierarchical predictive coding, this is the classical view as I showed you. So maybe we could add to this classical idea the orbitofrontal cortex, and that you have a detailed aspect information processing part, and you have a global information processing part that can aid the reduction of prediction errors. So a little bit more speculative thoughts about the state representation of global low spatial activation patterns. So my idea is, my hunch is that only one state representation is present each moment. And we can go back to this in the discussion, why I think this. That state representation in orbitofrontal cortex can prevail for a very long time, but it can also change very quickly. But it's about all or nothing, kind of. And that these states are hippocampus dependent, and also that ventromedial prefrontal cortex and RACC that we discussed before in the placebo studies serves as an interference between the orbitofrontal state representation lower level processes. I mean, just to mention, because of course we should think in networks. I mean, this is a lab work in connectomics, so everything else would be wrong to say. And we try to expand it a little bit. The ventromedial prefrontal cortex is a super interesting region in this. So you remember the relation it has in placebo, but it has a strong connectivity to orbitofrontal cortex. It's a part of the default mode network, and also a part of the Global Workspace Broadcasting Network. It is involved in self-related processes, for example, autobiographic memories. Ideas are that it's involved in kind of computing the self in the brain. It's involved in also regulation of self, emotion, and pain. So that is, I think, that is similar to what we saw in the placebo literature. Also, it's involved in evaluation that aids decisions. So it's this idea about a common currency that is computed by ventromedial prefrontal cortex. And it's active when context influence low-level processing. And the hippocampus and entorhinal cortex is really, really interesting. Of course, this is these regions are involved in learning and memory, but they are also involved in establishing cognitive maps. And I think it's really super interesting with the studies that Tim Behrens have done about they are involved in organizing conceptual knowledge in a grid-like code, which is exactly with these two other regions that I discussed before. But going from these ideas about place cells and finding your way in a labyrinth to general cognition, basically. And it's active and context influence low-level processing as well. So these, once again, these three regions are active there. I just have to make an so these regions seem then, I think they're really important in working together in this network. Just kind of, I wanted to just say a little bit about the theater I went to 2 days ago, the Burnt City. Has anyone seen this? It's called Punch Drunk. It's a theater company that usually they can, for instance, rent the whole factory, all the kind of maybe very old buildings, and they have like 20 different scenes. They have some rooms are extremely big, some rooms are small. They have and the theater is happening all over at the same time. So you are the active person in this theater and trying to build yourself an idea of what is happening. So you can walk after, and everyone has a mask actually. So you don't. All the participants, there can be like 500 people there, and you can walk around and see different aspects and try to build the story yourself. But I felt when I was there, this is really about context. I really to build up a complex context about the world. So I felt it was very similar to the story I tried to tell here. All right, so let's go to psychosis and what I call the psychosis paradox. So how should we understand what happens in a psychotic event? That's, I think, of course, if you haven't had one, it's really, really hard. But there are still things that can be, that we can note from our patients. So we can see that these individuals often have difficulties to focus on one stimulus at a time, especially in acute psychotic state. Instead, attention is often quickly shifted between different irrelevant stimuli that are perceived as highly salient. And the same individual may simultaneously have a set of delusions that are resistant to change despite being extremely unlikely or even bizarre to most patients. So you have both this very kind of jumping around experience and this very overly stable experience. That's the paradox. So we would say that the low-level system is noisy and the high-level system is overly stable. Okay. So how is this noisy system experienced? Well, I won't go too much into these studies, but using like a different electrophysiological, like EEG-based studies, for instance, you can see that if you have a stimulus that is the same and you compare it with a deviant that is similar that is not the same, so a tone that is different, for instance, you will react on that quite, or your EEG will react on that quite a lot if you measure that. However, this reaction is smaller in people with psychosis, but also their also their relatives, and those who are at risk and will later develop psychosis. And what you have to remember when we say that this is smaller, it's not the general, it's not the kind of the EEG that is smaller in general. It's the comparison between the deviant and the standard that is smaller. Here you see the random dot kinetogram. That's why I spent some time to tell you what it is, because I will show you a couple of times. Here you see the random dot kinetogram again. And if you don't use any type of instructions to the subject, you just show this. And basically you show it for 0.6 seconds, then there's like 0.8 seconds of blank screen, and then you show it again. Most people will at the second, when they see the second presentation again, they will experience the same rotation as the one that ended last time. However, sometimes you don't. And how much you do this or not, that is kind of how your perceptual stability is defined. And what you can see is that the more delusion proneness you have, the less perceptual stability you will have. And also, similar thing has been shown for schizophrenia patients as well. Reward learning is often shown that it activates reward-related areas less, for example, in anticipation of rewards. Fear conditioning also shows less, patients with schizophrenia have less fear conditioning than other patients. And actually the skin conductance is larger to the wrong stimulus, so to the CS minus. So all this adds up actually to that these patients, they are not good in comparing low-level stimuli to each other. It doesn't work. And the idea here is very old. Like I think Shichi Kapoor has summarized it well, that it's like a barren salience. So you experience salience for things that should not be salient. So, for instance, usually I say if I can hear, now I can't hear, and I hear I have that. I hear your computer, you're writing, and I didn't hear it before, but now I actively listened. If I would have been psychotic, that would have been a really important noise for me, possibly. So that's a barren salience. So I would make things that are not important. So we combine this with the ideas of predictive coding, and basically we land in this. So you have the idea is that the expectation that priors are very imprecise and non-stable, while the error signals become overly active. And the question is, why do we have noise in the title of this speech? Well, because I mean these types of error signals are really important for the brain to, as we said before, to learn what is around us and how things are ticking. However, what you can think about that happens in the patient is not that just one thing is wrong, it's more like this. So, because if you think about all the stimuli that are compared on a low level and are not compared properly, things become very important, others not. I mean, this brain is very noisy in that way. And this is something that the brain tries to work hard with to reduce this type of noise, because it means we don't understand the world normally. Okay, so we have an overly stable high-level processing also in these patients. This is what we call delusions. And there are two fundamental aspects of delusions and overvalued beliefs. One is about acquiring novel beliefs. The other one is about holding on to old beliefs. Okay, we'll just talk about acquiring novel beliefs first. This has not been well studied in cognitive neuroscience, but this is a very important thing, because people with delusion proneness and people with psychosis, they acquire new delusions. I mean, they were not born like that. And basically, we studied this, for instance, when it comes to COVID-19 conspiracy ideas, and we showed that, and others have done this as well recently, that the more delusion proneness you have, the more COVID-19 conspiracy ideas you will also have. But what we also now shown is that this is basically the only trait which if you're healthy, which determines if you vaccinate or not. And not only that, in those who have vaccinated, there's a relation between delusion proneness and the length it takes, the time it takes to vaccination. And basically using using a path analysis, we show that it seems that you have your PDI ideas, you develop your conspiracy ideas, and then you don't vaccinate. So there's actually, you take on new ideas and integrate them into your priors. And when it comes to predictive coding, I actually even won't go into this one. I'll show this one instead. When it comes to predictive coding, the idea is that you will have your aberrant salience in the brain. And you try to contradict that with a little bit of more priors to reduce these errors. However, what we say here now, given this whole kind of introduction I've had, is that the global system may be very important for this as well. So basically it can help to reduce these errors. So basically you have these aberrant salient errors, and you can also use the global system to reduce them. And this is basically what we've been working with for, I don't know now, almost 10 years. And as a lot of this work has been done together with Philip Sterzer, who was in Charité before, now is in Basel, and Katharina Schmack. She's the fantastic person who did that science paper. She was the postdoc at that time with us, and basically we could show that in patient, or actually in delusion-prone subjects, the more, I showed this before, the more delusion-prone you are, the less perceptual stability you have. However, if you do this manipulation, placebo-like manipulation, you see the opposite thing. So the more delusion-prone you are, the more you react to this, the more you actually use your higher order priors to experience the illusion. And this is not only how people rate. We use multi-voxel pattern analysis to show that the brain actually reacts like this as well. So it's not only that you're reporting as seeing that the rotation is to the left, the brain is actually reacting as the rotation is to the left. But what is more important, I think, is that we could also show that every time you switched, there's an orbitofrontal cortex activation, and it impacts V5, which is important for rotation and movement. And this impact, so this functional connectivity was larger the more delusion prone we were. Similar things was done in people with schizophrenia. So sadly, these patients had already, some of them were chronic and some were on medication. We didn't see a difference in how they rated, but we did see that the more delusions they had, the more positive symptoms, the larger was this effect of the manipulation. And we once again saw the interaction between the orbitofrontal cortex and V5. All right, just really, really quickly, I will show our studies as well here that we've performed the last years. So we tried to see if we can generalize this using the instructed learning paradigm that I showed you before. So basically, we showed four phases. Two had a narrative. So for instance, so for one, it could say that this person is known to be dangerous and has repetitively been convicted of assault and has a psychopathic personality. Be aware, he will give you electric shock. Or this person is known to be kind and empathic, this one. He has never hurt anyone, and he has always helped people who are in need. Relax, he will never give you a shock. And then two phases with no information. And basically what we could show is also quite interesting, because the subjects with high delusion proneness were more intensely kind of integrated this more than others, and rated more. When we looked at likability, that rated more larger difference between CS plus and CS minus phases for the instructed phases than the control subjects. And once again, we observed that the lateral orbitofrontal cortex was involved and had a similar type of functional connectivity as we observed in the RDK studies. This was shown also during extinction, so a completely different phase. So orbitofrontal cortex seems to be more involved in these types of paradigms, in delusion proneness and psychosis. And it's in line with that we actually, or this phenotype actually experience the world more with this prefrontal aid, so to speak. Last part, holding on to these beliefs. So we need not only to integrate them, but we need to hold them on as well. And there has been some beautiful studies using a paradigm called BAID, bias against disconfirmatory evidence. Has anyone heard about it here? No? Morten has, of course. So basically, in this paradigm, you can have a story or you can have a picture set. And you show just a small part in the beginning. And you should say, so here in this picture set, you should say, is this a toad or a lemon? What is it? Anyone?
Lemon.
It's a lemon. Of course you should say lemon. Okay, lemon, yes. But then you get more and more, and you should say what it is, and you should rate your conviction. In the end, it's a toad. So you were wrong. Sorry. The thing is that if you have psychosis, if you have schizophrenia, if you're delusion prone, you will hang on to your idea much longer. Well, you just had one photo, so we haven't tested the full thing. We could do that afterwards. So we wanted to use this idea but in a completely different setting. So we looked at extinction, fear extinction instead. And basically, because this is we know the neuroanatomy, the functional neuroanatomy of fear extinction quite well. We know, for instance, that the medial prefrontal cortex is involved in fear extinction. And basically, what we could show was that there was an interaction in this extinction. So this is just a likability score. So while the controls experienced more fear extinction, so less effect of the fear learning or fear conditioning, the other group, the high delusion prone, they experienced more fear after extinction, which is really weird. So they even tended to increase. And what we also could show was that this region, the medial prefrontal cortex, was more activated in the low delusion subjects than in the high delusion subjects and had a lower functional connectivity. So there is something to this. And the question is only, is schizophrenia general or a specific disturbance? So the problem with schizophrenia is there's so much happening in the brain. So if you look at chronic schizophrenics, you will see that virtually all parts of the brain are shrinking and different. So you can't really say anything by looking at these as these patients, I think, and not only the disorder itself, but also the different drugs changes the brain. So what we instead have to do is to look at the kind of early on, what is actually happening in early on, and before they get the psychosis. And basically, so if you study clinical high-risk subjects, it's really interesting because these regions that we discussed seem actually to be different in at least in their morphology, in their thickness, for instance, than in controls. And also, if you compare converters to non-converters, you see this as well. And we've done something interesting here because we use the genetic risk scores that I told you before, both for bipolar disorder and schizophrenia. And then we correlated that to patients with bipolar disorder, but also to controls. And basically we could see that in both groups you have a correlation to these regions. This is schizophrenia down here. And you see you have both the medial orbitofrontal, ventromedial prefrontal, but also the orbitofrontal, lateral orbitofrontal. And the cool thing is that if you look at controls and patients independently, you see that both groups have had this relation, and this is the overlapping green. So can we use connectomics to test our hypothesis? Well, I hope so. And this is really, I hope, a future collaboration to try to understand these processes from these models and other models that we have about brain function. I mean, we have already done a little bit. So this is the same study as I showed you before with the bipolar patients. And basically, we could show that using by using bipolar patients and also relation to genetic risk shows that the hubbiness of ventromedial prefrontal cortex is lower in patients than in controls. And this is the same that showed lesser thickness as well. And this study was made here by Henrik Fernandez, and it also shows that the provincial hub, now using the prevalence of functional resting state connectomics, this is structural connectomics, but basically showing that similar regions don't have a provincial hub as in the other one. And finally, we've also tried to use the LADA approach that is actually showing different states on a functional way, how you change between different brain states. And we can show that we were able to show together with Christine and Morten that the more preoccupation you have on your delusional ideas in delusion proneness, the more switching you have between these states. And also that this region, where this network where lateral orbitofrontal cortex is activated, has a lesser state probability in high delusion proneness than controls. So there's still a long way to understand the mechanisms underlying the experiences. But in the end, I want to show you, I mean, I think half of you have seen this, but I still need to show this. Have you seen this, anyone? No? Yeah, okay. So this guy is the psychological, this is the guy, oh, sorry. The old man, he's the subject. The other guy, he's the psychological tester. And he kind of asks, where can I find this street? And now they switch. And you see the old man doesn't see that at all. He looks at him and everything, and he doesn't know that he switched. And you can do this actually. I mean, the more strange you do it, you lesser will see this. But you can actually use change a man into a woman, or a white man to a black man, and some people don't see this because their priors are so extremely strong that this must be the same person. So so they are suppressing all this input that says this is wrong. And I would like really in the end to. I don't want to thank you normally, I just want to do this, and you can interpret it however you want to. All right, if you're interested in placebo, you can buy it on the net. This is I especially like the anti-monster spray, it really works. All right, these are all the most important people for the work I've showed you today. Thank you very much.
Thank you. What a treat. So, do we have some questions? I'm sure there must be lots of questions. What were they doing in the end? No.
We'll talk about that.
Yeah, well, what a fantastic talk. I mean,
Thanks.
Such a tour de force, it's very impressive. So many bits, but I'll just maybe pick a big picture idea that so it seems that in a sense what you were sort of hinting at is really that the orbitofrontal cortex is this kind of rough and ready global picture of the prior of what's going on.
Yeah.
Sort of a rough and ready multisensory gestalt, this is what's going on.
Yes.
And that's influencing, helping other regions form these views. And and I'm just thinking about a normal brain now, that this would be very helpful. Well, that is the part that is, in a sense, compensating for the massive increase in noise coming up through the prediction area.
Exactly.
So you've got these and it gets it wrong because the underlying data is wrong. Is that right? So is that have I got the right picture?
Yeah, you got the right picture. I think it's so basically on a more, let's say, it doesn't have to get it wrong, but it will use more any type of higher order explanation to interpret data. So that is why I think the let's say placebo-like effect is stronger in this phenotype. But if you stumble on something that is completely strange, you will also integrate that as well. And that could be, for instance, COVID-19 conspiracy ideas. So you will not kind of you will use all bits and pieces to reduce noise you have. And in relation to COVID-19, I think that is a good example, because I think that you have a lot of, already there you have a lot of, when you have to deal with these things, you will have a lot of lower level errors that are building up, that you need that are relating to this as well, and that you need somehow to explain. So why is that needle so horrible? So I've heard someone, maybe also on a more negative way, like we have all this, there's a reason why we get paranoid. It's so simple. I mean, it's the first delusion people usually show. So maybe more negative, so you interpret in general more negative things and need to explain that in a way. So things that threatens you, that are maybe a danger to you. That's the idea.
Right.
Yeah.
So the question that I have is like, why aren't the prediction? I mean, so you've got these massive prediction errors then, really even bigger ones, especially when you've got this delusion coming in, and then you kind of see something that's definitely like a frog, but you can't see it as a frog. So the question is, why are these even bigger prediction errors than normal, presumably? Why are they being ignored? Is it because there's so much noise that all of those prediction errors essentially are treated with very low precision weighting? Is that the? I mean, because this is the thing that you've got huge prediction errors in schizophrenia, and I know they have noise anyway, so it's maybe that all noise is ignored.
I think what one has to think about is that this is also something that it's not only momentary, it's something that builds up over a long time. In like, if you study people who eventually get schizophrenia, they usually have a prodromal phase, which is sometimes 6 months, so it's very long. And in this phase, a lot of them complain that things have changed, things are different, the world is a new place. Things are often negatively bad. So it's not something that is happening, during a minute. It's something that happened during months. So they probably walk around with a lot of prediction errors. And they need to build up something that they can handle these errors with. And then they come to something which is classically known as the psychotic insight. It's like, yes, I got it. And now I understand why the grass is so strange, or why or why someone behaves so strange. So it's all linked, of course. And so they build up this whole kind of delusional story that will explain all the prediction errors, basically.
Right.
So that's a way I think one should think about it.
That's very helpful. Thank you.
Yes.
Hi, thanks for the talk. So there was one bit that was about placebo in healthy people that I wanted you to elaborate on, because it seems evolutionarily completely counterintuitive that you would change sensory data to fit your generative model as
Mhm.
opposed to the other way around, right? Rather than updating the model because you have this pain signal,
Mhm.
the brain would just modify the pain signal
Mhm.
and pretend it's not there so that it fits your model.
Mhm.
And that in other cases, you wouldn't do that because, I mean, you can minimize your prediction error that way. You
Mhm.
can make it zero, right?
Mhm.
You just say whatever your model predicts, that's what my sensory data is,
Mhm.
and you modify it accordingly. And like evolutionary seems like detrimental to just modify sensory input to fit your model.
Mhm.
And why would we have evolved to do that, especially in the case of pain? Like, would the brain evolve to ignore a pain signal
Mhm.
just to minimize its own error?
I think it's a very good question. I think maybe a solution to that is to think about these 24 reflexes that I showed. I mean, I mean, if we would not use the models to change how we experience the input, it would only be 24 reflexes and nothing else, right? So we have already there, to a certain extent, changed the input to fit the model. So I think that but having said that, you can discuss pain. I mean, it's a harder, more tricky maybe to discuss pain in the same way. Why do we change? Why does pain change differently? Well, pain maybe has a meaning, and one of the meaning is that you need help. When you have help, maybe you don't need to experience as much pain. I'm just speculating now,
Yeah.
but that could be one reason why it's evolutionarily good to do that. But I agree. I think it's a question that deserves more kind of in-depth thought in general.
I mean, when you say like there's a difference between changing sensory, or I don't know if there is a difference between it at the neuronal level, between changing sensory input
Yeah.
and interpreting sensory input.
Yes, I agree.
Within a context,
Totally,
right?
totally.
You don't actually see people on those dots. You just like you're still seeing the correct sensory input. You're just making sense of it in a different way. Whereas like not feeling pain when
Yeah.
you're being like operated on is like far more of like just rejection of the sensory
Yeah, but
data.
there's a big, in the placebo research community, there's a big debate about that. Do we really change information processing from the pain signal, or do we just change the interpretation? And there are, there's, I mean, Torweger and colleagues have shown several studies that indicate that it's more the interpretation that we change.
Yeah.
At the same time, we do activate our opioid system. So if you give people naloxone, you will have less placebo effects, so opioid blocker. That has been shown several times, many times actually. And so it seems that both are possible. So I think the story the story hasn't ended yet. We don't know actually exactly how it is, but my gut feeling is that you have both parts. So you have I mean, the whole kind of, if you just look crudely on the kind of like the ideas from predictive coding and Bayesian inference and so on. I mean, it is more the interpretation that changes, but it's but the activation of the opioid system is more like that you actually change the real input.
Mm-hmm.
So maybe both exist.
Yeah.
It's just a one finger on this point.
Yeah.
Actually, within this active inference framework, you either, if you've got a prediction error, you either reframe your perception, or you do an action, and then essentially the release of opiates, endogenous opiates, are an action signal, and to reduce the prediction error, and it's all done within the same framework. So I would say, like, it is very much within the active inference answer.
Yeah, yes, in a way, and it's really active inference.
Yeah, this
exactly.
is the point of active
Yeah.
inference.
Yeah.
That you
You're activating your opioid system is active
it's an
inference.
action. It's an action.
It can be seen as an action.
Yeah.
Yeah, you're right. That's a really good response. Thanks. I will use that next time.
So, I mean, I think, if I may jump in here, I think what you told us about is about belief, but it's also very much about meaning making. It shows how we are these meaning making animals that cannot help even dots to see some kind of meaning, some kind of purpose to these things that are happening to us. And of course, sometimes that goes wrong. Sometimes we have a problem with noise that suddenly becomes salient, that becomes meaningful.
Yeah.
And because we are these meaning-making creatures, we cannot help ourselves doing that. Now, as a psychiatrist, of course, you know there are various drugs that you can give people so they can minimize the amount of major errors they make about doing this. Do you see much on the horizon that might be able to help give more meaning to these patients? I mean, what is your feeling about new treatment methods based on what you told us today?
Well, I mean, we are, I think, compared to other medical professions in psychiatry in general, we are so behind. But I see some light in the kind of tunnel, at least. And there are a couple of different directions. One is more that, I mean, we still don't know, actually, because no one has done, and it would be considered to be unethical to do a proper study where you treat patients with psychosis. Half of them you treat them with antipsychotics, and half of them you do nothing, and see how the development of the progression of the disorder is kind of taking place. But the hunches and the indirect data suggest that actually it helps to block the psychotic symptoms, which is very, I mean, as I've shown you, this is very much down the road that we're blocking it. We're not blocking these initial things that are happening, but it still seems that there's a kind of a feedback system. If you have these psychotic experiences, that it will be worse. You can think of it maybe in terms of learning, that you learn these delusions more, and if you block them, you don't learn it that much, and you can unlearn more easily. So I think that early treatment is really, really important. And there are also data suggesting that psychological interventions, where you actually work with your thoughts and your overvalued beliefs or delusions, can also kind of stop this progression, or at least make it slower. So that is one part of it, and we need to understand this much better. There's a lot to do. We are a lot behind, but at least there's some hope there. The other part, which is huge now, is where clinical tests have started, is to attack the immune system, and that is really, really cool. So we are actually, there are several studies I know of that are starting now that are using similar monoclonal antibodies that are used in MS, for instance, that are tried out in patients with psychosis. And that's so that is going on the up the river. This is down the river, this is up the river. And so there are hopes for the future for these patients group. But I think really early interventions, finding these patients early and starting up all the interventions there can change the progression.
Great. Have we got any questions online? Well, I think it remains.
I have a question.
Thank you again.
I'm sorry.
Oh, there's a question. Please.
Thank you so much. Well, thank you so much for the talk. I have a question that, well, kind of the psychotic patient that you're talking about, it's a deviation from the normality, kind of. I'm kind of wondering whether there is kind of, if you look about kind of the other side of deviation of like a genius brain, whether there is any connection, or like whether they look similar, or whether it's a different kind of connectivity in the brain, or like, because a lot of things what you're talking about is that genius is so cold, right? Can see things that other people don't see, or they can connect things, or like the psychotic insight as you talked about, it can actually sometimes be something of a genius mind that can lead to some sort of, you know, discoveries or new ways of looking at the world. So I was wondering, maybe you can talk a little bit about that, whether you have looked at those things.
I mean, so this relation in between creativity and psychosis is well established. So I have a colleague of mine, Simon Kiaga, who did epidemiological studies on 100,000 people or something. I don't know how many, something like that. And basically what they showed is that So if you're a depressed patient, you will not be creative, and and the family doesn't tend to be more creative than average. If you're bipolar, both you and your family will be more creative on an average level. If you have schizophrenia, you will not be more creative, but you will not be less creative, and your family is more creative, which to me actually explains that these people are actually extremely creative but handicapped. So in terms of that, in that thinking, I think if you think about the trait that exists in the general population, these people who maybe have some weird ideas sometimes have the finest and coolest ideas of doing something new that no one ever thought of. They make an association which we would, that a more average would never do. So there is a benefit of doing this. However, what the benefit is of the other side of this distribution, I think it's really, or maybe not the benefit just, maybe there's a handicap as well. I think we have no idea. It's a mystery still. So that would be really cool to see. I mean, we've done this with for instance, cognition and executive functions, where we've looked at both sides of the normal distribution, but here we actually don't know.
So I love that. We end the talk with a mystery. Yet another mystery. Thank you again.
Thank you.